about I'm going to talk to you about research at Mount Sinai, which is in New York City. The title is From Samples to Population, Interactive Visualization of Multi-Omics Data at Scale. Let's see why it doesn't go. Okay, so before I start, let me give you a little bit of information, background information about our research group. We are a computational cancer genomics lab, and our aim is to identify and target drivers of cancer genesis, progression, and therapy. And this is a word cloud of some of the words mostly used in our abstracts, in our research, and you can see cancer, tumor, but at the same time, you also see network and yeah. And while we're attempting to answer these questions, we develop software and web interfaces. Some of the tools that we have developed, I list here, and I'll mention briefly Primavo. And we collaborate with clinical, experimental, and computational groups such as the NCI Clinical Proteomic Tumor Analysis Consortium, the CMAC-CIDC Immuno-Oncology Biomarkers Network, and until recently, the NIH Human Immunology Project Consortium. So within these efforts, what we try to do is to build bridges between data visualization and cancer. And what that means is that we have to talk to a lot of people from different backgrounds, meaning from clinical oncology to web development and visualization design. But in the end of the day, we do have questions that we want to answer in how to better understand cancer mechanisms, how we can identify targets and biomarkers. And so all of this goes hand in hand with basic research and computational oncology. So within these last few years, we have been developing a tool called Primavo, Precision Immunology Monitoring Analysis and Visualization Online. And this was thanks to a Cancer Moonshot grant from the NCI, from the previous President Biden's time. So the question is, who will benefit the most from cancer immunotherapy? And we want to be able to answer this question. So if we have a group of patients that have advanced cancer, the question is, who is going to be in the group that is lucky enough to have complete response or partial response, and then on whom the therapies are not going to work at all. So maybe we should try to be able to identify the markers so we can better identify who should be screened. So this is part of the questions within the CMAC-CIDC, which is the Cancer Immune Monitoring and Analysis Center, CMAC, and Cancer Immunology Data Commons, which is CIDC. And what these consortiums are doing is that they're generating disparate immune monitoring omics data sets. So these are a large numbers of different assays, from protein analyte expression, gene expression, cell abundance, antibody levels, genetic mutations, microbiome, in situ characterization of immune cell infiltrates, and spatial sequencing and single cell sequencing. And not only are these assays diverse, they're also measured with respect to time. So there are multiple time points, but that include baseline immunocompetence and changes during the treatment, and also what happens to these markers once the treatment is over. So what we aim to do with the tool that we have developed called Primavo is to have all these data sources from CMAC-CIDC in addition to any additional data that we have, and these data types into a portal that makes it easy to access and explore. So the data types are from the assays that I listed before, and this is all in the Primavo backend of our web tool to visually analyze and interactively explore these assay results. And in the front end, what the user sees is just study selection. So the clinical trial of interest, and there's like a summary of the clinical trials. So the user selects or searches for the trial of interest, and after study selection, the users select the data types they're interested in, so they can filter the patients and the clinical attributes and assays or treatments, any assay that they're interested or patient subtype they're interested, they can filter for those. And then they can explore these data sets using different interactive visualization tools such as clustering or dendrograms or other visualizations such as scatter plots, bar charts, volcano plots, pie charts, and I'll show you all of these. And in addition to those, Primavo has tutorials, user feedback, and FAQ sections. And so we wrote Primavo in the React framework, and the backend uses Python, Django, and yeah. So it is not available due to data access restrictions. However, we just recently gave NCI CIDC portal where it's going to live access. So we've already delivered the code to the NCI. So they will be hosting the tool at the NCI hopefully soon. They already have all our code. And in terms of the study selection page, this is where we list the cancer immunotherapy clinical trials, and the user can select any of the trials they're interested. The samples per assay is listed, the assay type, the study name, patient data, study enrollment, and then they can also go to clinicaltrials.gov to see additional information on that clinical trial. And once they select the clinical trial, there is a data selection filtering page. And this page includes all the data that is available within that study. And it includes therapies, assays, cancer types, response rates, mutated genes, stage, treatment, drug, sex, adverse events, everything. So we have the therapies. So this is a snapshot of the data selection page. And then if we were to do a video of how we interact with it, so we can see the selected number of patients and the total number of samples on top right. And then the therapies here, we have 3 different therapies with different drug dosages and escalation of the drugs in the specific clinical trial. And the user can choose any one of these, and everything updates in real time. And here we have the assay types, the cancers that the patients have, and here we see complete response, partial response. So if we want to just look at the people with partial response, we just click on the partial response box, and then everything updates in real time. And let's see. And we can also look at, for example, different demographics. If we're interested in individuals with European ancestry, for example, then we see that how many samples there are and how many individuals there are. And we can also clear all the filters. And we can provide specific age cutoffs if we want to look at, for example, only people younger than 42 or older than 42. We can do that, or any arbitrary age cutoff that we want to have. We can choose from this from the interface. So it helps us subset our cohort really well. And in addition, we can also look at survival plot, so KM survival plot, so that maybe we want to only look at data related with people associated with a specific treatment that have good survival or bad survival. So we can choose the cohort also based on survival before we go ahead with the subsequent analyses of the data sets. And here we can, for survival, we can look at the survival in female, male. So it gives us like different ways of interacting with the data that were just in tabular form, and to visually interact with the data on the fly so that we can generate hypotheses or study them a little bit more in detail. And from the, let's see, yeah. So from here, we can go to the data exploration page where, for example, if we choose the site of assay, in the left axis, in the x-axis, we see the markers that have been measured with the assay, which is the cell types, and on the y-axis is the de-identified patient IDs. And so this just gives us an idea of the assay results overall. And then here we can look at it in this additional heat map. We can look at the summary comparisons between different groups at the same time, so that we can see that there are statistically significant differences between certain groups, both in the positive and negative direction. And we can maybe focus on those. So here's, for example, another thing we can do is that if we have a marker of interest here, for example, CD4 T cells on the x-axis, we can just click on this marker and then see how it changes with respect to time. And then we see a box plot of that marker and how it changes with respect to time and treatment in a specific treatment. And so if we do a demo of that in the data exploration page, and you see we have site of serology Olink, so the user can choose whichever assay they're interested on the fly and then interactively explore the data sets in that assay. They include Olink, RNA-seq, WES is whole exome sequencing, and yeah. So here for CyTOF, which is the cell frequencies, the other thing is we can also we visually explore metadata. Here we chose metadata, treatment, sex, and race, but we can change those metadata based on what we're interested in, and then again update that on the fly in real time. So everything within this heat map is completely interactive. And here we can also cluster different groups based on their values. For example, here you see different colors corresponding to different clusters within the markers. And for example, we can also change and cluster them with respect to sum or variance, and then the whole heat map updates in real time. And then we can, look at the different, for example, for these cell types, we can look at their origin, parent of origin, which is myeloid, for example. We can see that we can make these clusters more finely grained or more coarsely grained, depending on what our interest is. And actually, there are different parent classes, and we can even put all of them together to look at all these cell types together. And we can order them with respect to just sort them with respect to cell type name. And we can also similarly order the columns, and the columns correspond to the patient, so we can order them with respect to, again, cluster sum, variance. These are some of the things we can do, for example. And then we can click on treatment, and then it reorganizes the heat map on the fly based on the treatments. So these three groups of treatments we can see. Similarly, we can do by sex. So, I mean, it just makes data exploration much easier and much more intuitive through this interface. So again, here we see race. And let's see, what else do we have? Yeah, and now we can also cluster the individuals based on the data. And then if we look at the details, we can see the details, the metadata details of each cluster in terms of race, sex, treatment, and the percentage of each individuals within each cluster. And then we can make these cells more. We can also make them more dark, more the colors more pronounced or more lighter, depending on what our interest is. And what else here do we have? And we can also search for specific markers. So the query box helps us identify, for example, CD4 T cell. If we're interested, we can search, and then we can make the heat map more opaque or less opaque, depending on what our interests are. Sometimes we want to be able to do that. And let's see. And I did these videos a while ago, so that's why there's sometimes disconnect. So when we make it less opaque, we can see certain really extreme values much easier. But then we prefer to have like medium opacity so that we can see in between values also. And also we can, from the metadata on top, we can remove some of the metadata and add additional ones. For example, time point, we can remove and add a different type of metadata there. And also we can z-normalize the data, and once we z-normalize, we can look at the z-normalized marker values. And okay, once we select, sorry, too fast. So once we select the CD4 T cell, this is the box plot I showed you earlier in the previous slide. And we can see how CD4 T cells change with respect to race or sex or treatment. But if we want to look at a different marker, we can just look at it from the box plot that we have. And again, everything updates in real time, and we see like different time points. And here, what we see is, for example, the patient groups in terms of complete response, partial response, and how this marker changes over time in these different groups. And of course, our number one interest is how can we better understand who's going to show complete response or who's just going to progress. So those are the main questions that we want to be able to answer, and if that marker is going to shed some light into this, so that we can in the future be able to predict who's going to be able to be responsive. And here we are looking at different markers and how they change over time between complete response, like between responders, which are brown, and like others, which are pink. And so let me see. Yeah. So that's it with this video. And let's see, we saw this one. And the other data set that we have is the summary analysis. And here we are just comparing different analysis results. And and once we have the summary analysis, we can look at any comparison. And for example, partial response versus PD in the y-axis, and then like progressive disease, and then we can have a volcano plot of how the marker values in the partial response, which is good, versus progressive disease, which is bad, and then see which markers are actually useful and are actually good markers that we want to see in a patient, and which ones are the ones that we don't really want to see because they are suggestive that the treatment is not really working very well. So this is the summary heat map page of the site of results. And here are the comparisons. So basically comparing baseline complete response with progressive disease and so on and so forth. And here we can see which ones are actually not really not really giving us any information, versus those that are statistically significantly different between responders and non-responders. And here we can also, cluster these responses also on top, and also do the opacity slider. So we want to look at the more extreme cases. And we can also change our threshold so that our p-value for differential difference between partial responders and non-responders, maybe we want to see subtle changes, or maybe we want to look at, based on the p-value slider, just the most significant changes between them. So depending on what our needs are, we can update. And also we can look at the log fold change value between them, or log p value between them. And again, all of this is updated in real time on the fly without any issues. So and then if we click on any of these comparisons, for example, in the previous slide I showed partial response versus progressive disease, then we get a volcano plot. So these are positively associated between the difference. So these are probably good markers, because we want to see them, because these are high in responders. But on the other hand, the blue one are the ones that we don't want to see in a patient, because yeah, they are negatively associated. So that's it in a nutshell with the data exploration, the summary data analysis. And let's see. Oops, stop. Okay. In addition to these interactive heat maps, we also have interactive oncoprints for whole exome sequencing data. And so here, for example, based on what mutations that the patient has, we can have, oops, sorry. We can have for example, the P53 mutation in the tumor, or P10, or BRCA1 and BRCA2, and then we know if it's an in-frame mutation, missense, truncating, if there's upregulation, downregulation, if there's messenger RNA information, or deep deletion or fusion. We can have all of this. We can interact with these in the fly using the OncoPrint visualizations. And another thing is immunohistochemistry data. So if we have slides from immunohistochemistry, and we can select certain regions of interest, and once we select the region of interest, we can look at them in detail in user-friendly plots, so that we can see which specific markers, for example, CD8, CD3, or the two of them together are here, or they're not there. So we can look at the region of interest in more detail, and we can pick up different regions from the slide, and we can do this for all the slides that we have. And so yeah. And here, let's see. Yeah, so we have the, if we are to demo the immunohistochemistry tile map, so we choose a region of interest, and we chose 20, and here we can zoom to this region very quickly. And then maybe we want to look at a different marker like CD3 or CD8, depending, or maybe the two of them together. And then on the fly, we get these, how these markers are on the slide. We can see them right away. We can zoom in and out. The resolution is dependent on the data that we had, so it's not a Primava thing. And then we can also see what percentage are positive on the type of cell that we have, the percentage of that cell type in a specific region that we are interested in. We can also look at that in real time. So now we chose another region of interest. This is region 37 in the same graph. And then we can look at these markers now, like different markers on this on this region and see what is the percentage amount of these markers in this region, for example, and how are they spread within the slide. We can also see these information. They're staining basically, and let me see. Yes. So, and then looking at the whole exome sequencing data in terms of the anchor print, genetic alterations, again, we can interactively explore. For example, there are a lot of truncating mutations on P53, and we can zoom into certain regions, a little bit more detail, not regions, but like within the cohort, we may want to see how which individuals have which mutations and what type of mutation and which gene. So this oncoprint part is still under development, but it gives us some information on mutation frequency and the mutation type, and we can also choose maybe deep deletion or amplification. Then it only shows in the oncoprint the mutations we're interested in. And here, every vertical line is a patient, and each patient can have one or more mutations on different genes, and also in the same gene, they may have multiple changes in their tumor. So we can see all of these in real time, and the gender, race, and other metadata we can also interact with on the fly. So we may want to see the women with P53 mutations that are truncating, for example, and then we can just, we have the cohort, we see what other mutations they have, and study them in more detail on the fly. So overall, Primavo is a web-based tool to visually analyze and interactively explore immune monitoring assay results from cancer immunotherapy trials. The results can be utilized for observational studies or inform subsequent preclinical studies and clinical trials. The users can filter, query, visualize, and share a number of high-dimensional assay data of study participants at multiple time points. And it's fully customizable and flexible with visualizations at both participant and cohort level, even though the cohort level is under progress, and it's fully customizable. And the design process in the development of Primavo, we followed user-centered design principles, which means that we met with CMAC CIDC investigators that are at Mount Sinai every week. And every change that we did, we talked to them, and we asked, does it work? Does it not work? How else can we improve it? And so we continuously solicited feedback from them. And then we solicited feedback also from in quarterly periods from the CMAC CIDC investigators group, and for its end users. So what I'm not showing here is that through this study, actually, which is focused on cancer patients, we started another study with a group from Harvard, from Dana-Farber, Elad Sharon's group. So he is very interested in cancer patients, but those patients have COVID-19, so vaccination. But after the vaccination, some of their markers change, and also when they develop COVID, their markers change, and their disease progression can change. So for that, we developed this tool, Primavo COVID, which doesn't have some of the data access restrictions that Primavo has, and that is publicly available. And I'm going to show to you at the end of the presentation the link for that. So that is going to be a separate study, for example. And all the investigators, different investigators groups within CMAC CIDC that generated data, we met with them one-on-one with their team members. Basically, we had several Zoom calls with these teams in order to make sure we understand their data correctly so that we can communicate their data correctly. So all of this involved a lot of communication or trying to communicate. In addition, so another tool that came out of all these efforts is cluster chirp, and it's a funny name, but basically we ended up spending so much time on these interactive heat maps that we decided that we could take a part of this and have it as an individual standalone tool, so people they may not have cancer immunotherapy data, they may be looking at something completely different, but in the end, if they have tabular data that they want to explore from different assay types, they can do that in a heat map. When we looked in the literature, it seemed there were a lot of problems with a number of the heat map visualization tools just because of the sheer scale of the data sets that are coming out from single cell sequencing, spatial sequencing, or just even when you do Olink, but you do it on like thousands of people, now you're talking about issues of scale. And however, you do want to be able to interact with these data sets in a way that you want to see the bird's eye view or overall view, and then perhaps dive in. So Cluster Chirp is a GPU accelerated web platform. It is AI supported, which means I will explain to you what it means. And so it enables interactive exploration of high dimensional omics data. So now we're talking about looking at thousands of individuals, or at least like in the end of the day, what Cluster Chirp can handle is up to 10 million cells. And the 10 million cells, just remember it's X times Y. So if XY times Y surpasses 10 million, I cannot guarantee that it's going to work, but it may not, depending on the data. But we have tried up to X times Y equals 10 million, and it seemed to work well and on the fly. So that is quite a advancement. Cluster Chirp is completely free because it's just an interactive visualization tool, which means that you can, from this link that I have here, clusterchirp.mssm.edu, you can explore it right away. And so basically, what we wanted to answer with Cluster Chirp is the computational bottlenecks, the complex user interfaces of some of the heat map tools that we see in the literature. We wanted on-the-fly clusterings and interactions, so we are addressing slow processing time. And another aspect that our users were very interested in was to have manual biological interpretation of the data, because what happens once you do a heat map cluster, when you have a heat map, when you cluster it and you see a group that seems to be biologically relevant, then you have to take it and put it into an enrichment tool, and sometimes you have to change their annotations. If it's proteomics, you want to change it to genetic gene names, gene IDs, etc. So instead of having these multi-steps, we said, okay, we can also do some on-the-fly biological enrichment. So we incorporated that. Its performance, it's very fast with these 10 million values. It's because it's GPU accelerated and it does a lot of parallel processing in the server. And another thing that our users wanted, and these are users that do computational biology, but they're more interested in the answering the questions, and people perhaps have tool fatigue in trying to understand different aspects of a tool. So instead of doing that, we aim to make it very simple. So we added a natural language interface so that people can just write what they want instead of use the mouse and the keyboard. I mean, they still have to use the keyboard. So instead of going through multiple steps, they can just interact with the chat box. And that's why we call it chirp, because it chirps with you, or you chirp with them. And yeah, so we also have pathway analysis, and I welcome you to interact with it as I go by in my presentation. So here is the flow chart of Cluster Chirp. Here is the user. It can also be a man. And so this is the AI chatbot that the user can interact with, or they can just, interact with ClusterChirp the classical way and input their metadata and data matrix. And once they input their data, um, they can do a number of interactions with the heat map, zoom, pan, sort, or cluster, crop, look at specific pathway, change opacity, etc. And they can do normalization or hierarchical clustering. So the visualizations use DeckGL, D3GS, and React Force Graph. And in the back end, we use Python, Django, scikit-learn, NumPy, Pandas. And the visualization output, we do have networks, and I'll explain to you why, and also heat maps, and we can download it in different formats or take snapshots of the heat map for publications. And this is a snapshot of ClusterChirp homepage, and it does have a lot of the visual elements of Primavo's SA heat maps, as I said. But as I mentioned, it's very high dimensional, and in terms of the number of cells that we have here, and we upload our omics data here in the upload data, and then, or we chat with the AI box here, and then we have the control panel here, and then on top we have examples. We're still working on FAQ and contact, and then we do the visualizations. So here is like a little video of its interface. So, and here we can take snapshots from this little camera, and yeah, so the opening page already has an example that users can interact with to get a better understanding of a heat map. And this is the overall heat map of the whole overall view of the heat map. We can turn this on and off depending on if we're interested in this little box or not. And here we upload the data, and that's it. So it's already uploaded, and this is now we're looking at the data that we just uploaded. And we can look at the metadata. We can cluster it with respect to sum. We could cluster with respect to rows. We can cluster with respect to columns. It just does everything on the fly, very fast. And it does tell you when it's done so that you know that something's happening in the background. You can look at each cell, and it gives you some cell properties. And in the on the fly view, you can move around so that locations within the main heat map through the little box. And then within every cluster membership, you can see the metadata and the details of the metadata in the fly. You can change the metadata you want. It's similar to how we have it in Primavo. And then these all change in the fly. For example, you can add histology and remove treatment or etc. And let's see, what else can you do? Yeah, so okay, so another thing that you can do is you can analyze, once you have a heat map, you have a cluster of the markers that you're interested in, you can, what you can do is you can look at them in a pathway view or in a network view. And another thing is you can search for a specific marker here, IL-6, for example, and then look at the vicinity of, look at all the information around this marker, for example, and basically interact with the massive heat map that you have. Again, you can change the opacity of the heat map so that you can see more subtle changes if you're interested, or you can make it less opaque so that you're only focused on very extreme values. So you can do that too. And okay, so in terms of the AI interface, there is a flexible command system. We did give some pointers to the users so that they know to guide them what they can do. Like it involves filtering, selection, sorting, clustering, search, and visualization. And so this way, the AI, the chatbot, gives control to the user beyond the standard controls, and the user can, define their own parameters, for example. And here now we are interacting with the tool through the chat box, just to let you know, if you say hello, it's not going to say anything back, but it's going to know that you said hello. And once you open a session, it keeps all the previous commands that you provided. It keeps it until you open a new session. And all of the work is happening in our server in the backend, which it has like security for like HIPAA. So there is no HIPAA violation. However, if you don't want the Sinai server to, and anything is saved temporarily. Once you turn off the session, everything is deleted. But if you need additional security, we have containerized cluster chirp so that you can just download the containerized version and set it up in your local computer. So there's no information transferred whatsoever. And then there's no possibility of data leak, but I mean, with the understanding that our server also has a number of security protocols for HIPAA, so it's the user's choice in the end. We can, as you see here, we can select different parts of the heat map depending on what we're interested in. So for example, we can say select only women, or select the top 10 markers, or anything that you write, it gives you away. Or you can just say, you know what, I'm interested in pathways, so I just give me only the genes in hallmarks, cancer hallmarks, for example. And then it's what it's showing here. Show cancer pathways, and then that's now you're looking at only the genes in cancer pathways. So there are multiple different ways of filtering the data that you have. And if this was proteomics data, we just, map it to genetics, Hugo ID, gene ID. But if it's other type of data, we may not be able to map everything, but we can always try. And if not, you can always reach out to us and say, how about this annotation? And we'll try to accommodate that. So that's the chatbot interface. And let's see. And another thing that we did is that once you cluster it, and now you found a cluster that you are very interested in, and you want to know more about that cluster, well, we can do a deeper dive into that cluster, and that we can do in 2D or 3D. So here we're looking at the same cluster, but in different ways, so that we will know within the cluster how are your markers correlated with each other, because it's not going to be a uniform correlation among cluster members, right? So now you have a cluster, but within your cluster, as you see, you have some subclusters. So so you can see how they relate to each other in the 2D view, or you can look at them in 3D view again, like have them in a circular layout, and try to see how they relate to one another. So the 3D is it does Leiden clustering for the subgroups, and the idea is to, in the end, in both 2D and 3D, to reveal additional gene relationships through the network analysis. And another thing that it does is that you can also pick a cluster of interest and do pathway enrichment, and then it will give you the enrichment score. Here it's very small, the p-value, and the TNF alpha signaling pathway, for example. And so you will get from genes to biology, you can do enrichment on the fly and query multiple biological databases through Cluster Chirp tool. And here we have a demo. In-depth clustering, you can do that. And or you can look at them in terms of networks. So we have and here, for example, you're seeing how these cluster members interact with each other within your cluster and their connectivity. And all of these are interactive, and you can, turn labels on and off, or do other things with it. And here it is. Like, you can also look at the overall how your cluster is located in comparison to all the other markers within the data set, or just the markers within your subclusters. You can do that too. And you can zoom in, out, etc. And then here you can also do, once you select the subcluster, you can also do enrichment on those, and then look at the p-values and the details and the members of those clusters also on the fly. And for that, you can do pathways, ontologies, like gene expression data, diseases and drugs, cell types, all of these you can do. And then let's see. Oops. Okay, so in summary, ClusterChirp is a web-based platform to visualize and interactively explore high-dimensional omics data through GPU-accelerated heat maps and clustering analysis. The results can be utilized for pattern identification and biological pathway exploration in research studies. Users can upload, cluster, filter, and analyze large-scale genomic, transcriptomic, proteomic, or any other proteomic data set with real-time performance. The AI-powered natural language interface enables researchers to perform complex visualizations through conversational commands, making advanced visualizations accessible to users regardless of computational background. And the design process follows user-centered principles, incorporating continuous feedback from biomedical researchers to ensure intuitive functionality and workflow integration. And as I mentioned before, this is where ClusterChirp lives right now. And so in summary, the tools I presented today, it's Primavo. It's soon going to be available at the NCI CMAX CIDC portal. We already handed off all the data to the NCI representatives. And we do have a beta version of Primavo, Primavo COVID, and this is the link. And it does have login access because it's not published yet, but once it's published, this is going to be publicly available. And this is in collaboration with Elad Sharon from Dana-Farber. ClusterChirp lives here. It's free. Feel free to use it. If you want to see any functionalities, if something's confusing, unclear, or you'd rather be different, let us know. If you also have an example that you'd like to share with us to use, we'd welcome it, and we could even give you credit because we're looking for good examples for our paper now. I did not show to you, we have other tools that we have, FOSNetVis, immune regulation, and CPTX CCRC, but this is where they live, and you're welcome to inquire about them if you are interested. And so the work that I presented today was the both ClusterChirp and Primavo. They were developed mainly by Oshorawal, who's a senior software programmer, software engineer in our group. And the initial version of Primavo was developed by Kevin Ulrich. We had Bark, Nitin, student interns who contributed to very early versions. Maya Sai Salwan, she's an instructor, she's a faculty now, and she was helpful in conceptualizing Primavo. So Sasha Ginyatic is our collaborator who manages the Sinai Immune Monitoring Core, Human Immune Monitoring Core, and a lot of the data that we showed here was generated in his lab. And both him and Edgar, who works with Sasha, we continuously solicited their feedback, and they have been both tremendously helpful in making the tool user-friendly. And this is where we are here, as you see. This is Manhattan. This is Central Park, and you see the peace sign. So we're, I guess there's some irony, peace sign in America. But yeah, so we are around here, so close to the peace sign. If you ever want to come visit us at Mount Sinai. And these are some of the consortiums we work with, and these are the NCI institutes that we interact with, and we love feedback. If you'd like to discuss, this is our lab GitHub. You can email me here, and also you can find me on LinkedIn, etc. So thank you. And with that, I'm happy to answer any questions you may have.
Thank you so much, Zeynep. Wonderful overview about these tools that I had no idea you were developing. I guess we're not that many, so please just unmute yourself and ask your questions directly to Zeynep. Don't post it in the chat. That would be wonderful. Um, Maybe I can like start with one general question that I had. So the dashboards that you've been developing, they will be handed over to NCI, right? The at least
Yes.
the first one. And what is the plan that they have with it? Is it going to be like the front end of all of their clinical trials? So as in, we better get used to learning how to use them ASAP because that's going to be our way to interact with this data, or what's the idea there on that side?
So it's going to be, so this is specifically for cancer immunotherapy clinical trials, the CMAX CIDC, and we already populated it with these data sets.
Mm-hmm.
So, and we will so basically they already have a website, like a web portal for this, but it's pretty much here's the table download. So
Mm-hmm.
Primavo is going to be integrated with that data set so that the users are going to be able to actually interact with the data instead of just downloading the table. So there's already a dedicated web address, so to speak, to it, and that's not open to the public. But what we are planning to do is we're going to containerize Primavo also. We've already containerized Cluster Chirp. We're going to containerize it so that for people who want to use it in their institutes and with their data or with their cohort data sets, they can just take it and, use it that way. Because one limitation that we had is it's a lot of work, but then data access restrictions make the tool also inaccessible. So that's why we are very excited about the COVID Primavo, so that we can show its functionalities and it's going to be completely free. And containerizing is another solution. So if in the end we wake up and everybody uses it, all the better. But right now, this is where we are. So
Thank you so much. Feel free, guys, to ask Zeynep. Officially, we have 3 minutes left. I guess Liz already said goodbye and thank you. So in case you didn't see that, Kari,
mhm.
hey. Mm.
Ah, yeah. Hi, Zeynep. I have some questions. First, I was wondering that when it comes to cluster chirp, there's a tool called Clustergrammer also from Mount Sinai. It looks somewhat similar. Are
Yes.
they the same tool or?
Yes. No. Yeah. Well, so excellent question. So we did use some of the design elements of cluster grammar.